Welcome back. The hair is finally on display. It's been long enough. Ramadan is over. Eid Mubarak to all the Muslims out there. Shout out to y'all. I have some bad news. This is the second to last lecture I will be giving. So when we come back on Tuesday, it will be my final lecture, and we will finally do lab techniques. We will finally go over separation techniques and spectroscopy and labs and things like that. So that will be my final lecture here at Brooklyn College until August, the next time that this series starts up again. So for everyone who kept up and everyone who's been studying for the MCAT, thank you so much for your support. Speaking of support, I have a Buy Me a Coffee link if you want to support the channel. It's not necessary, but you can. And I'm also going to put my Venmo down there. Some people have asked me to do that because Buy Me a Coffee takes out a larger percentage than Venmo does. So I'll put it down there. If you guys feel like donating, donate. Also, half of what I make from this channel every month goes to charity. Last month, we raised $45, and since it was Ramadan, I'm donating all of those $45 to Doctors Without Borders instead of half. So if you want to donate. You don't have to. It's much appreciated. And I hope you enjoy the lecture. So today, we are doing part 2, the long-anticipated part 2 of the Biochem Rapid Review. All right, let's do it. So Biochem Rapid, part 2. So last time in the Biochem Rapid, we talked about amino acids, we talked a little bit about the ETC, we talked about fatty acids, we talked about glycolysis, we talked about glucose, we talked about enzyme kinetics. This time, well, since we talked about fatty acid oxidation, obviously fatty acid synthesis, blah, 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 whatever. Now we know that for both of those, we actually sort of use something that we don't normally use in other processes. And what is that thing that we use? I don't know. Actually, no, I'm saying the wrong thing. I'm looking at the wrong part of my notes. So, my bad. Start over. Let's talk about one of my favorite molecules ever. Do you guys remember glutathione? Right? Glutathione basically becomes glutathione oxidized when it picks up antioxidants and pairs with itself, and basically becomes 2 glutathiones stuck together by a cysteine bond, right? What do we use to reduce that bond? Do you guys remember? So glutathione turns into 2x-glutathione. So it's basically G, and this is the way we write in medical school. When we're talking about it in medical school, it's 2GSH, so glutathione with the SH group, becomes GSSG. Right? So it's 2 glutathione molecules like linked together like that, right? So when we want to convert back this way, what do we use? We use a molecule of NADPH. And that makes NADP+. Now there has to be a way to regenerate NADPH in order to protect the cell from oxidative stress. What is the method of replenishing that NADPH? This is a very high yield concept on the MCAT. The pentose phosphate pathway. Replenishes NADPH stores. And the reason, does anyone remember? It's a very, very complicated and intricate sort of biochemical fact, but does anyone remember why the pentose phosphate pathway uses NADPH, but glycolysis, gluconeogenesis, and glycogenolysis use NADH? It's because you want to keep nucleotide synthesis apart from energy production and energy breakdown. Because if energy production, energy breakdown, and cell production, like cell turnover, nucleotide synthesis, if those were coupled by the same reagents, then they would both follow the same schematic. They would both move in the same direction at the same time. But there are certain times that you have to halt cell growth and keep churning out energy, and there's certain times you have to halt energy and churn out cell growth. So therein being, you have an NAD plus to NADH ratio, and you also have an NADP plus and an NADPH ratio. And these two ratios are independent of one another and regulated in independent processes. So that one doesn't affect the other, doesn't affect the other. Does that make sense? Yeah. Good. So that's what I wanted to say about the pentose phosphate pathway. And what's the sugar we put into the pentose phosphate pathway? It's ribose, right? ribose, and then it can turn into like ribulose, and there's like xylulose or something like that. Very weird sugar name inside the pentose phosphate pathway. All right, that was the PPP, also known as the HMP shunt, right? The hexose monophosphate shunt. Well, sorry, we put glucose 6-phosphate into the thing, and then it turns into ribose and blah, 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 whatever. Right. Let's talk about glycogen. What is glycogen? I mean, you don't know. What is glycogen? It is a polymer of glucose. It is a glucose polymer. Are all glucose polymers the same? No. There's glycogen, and there's starch, and there's cellulose, right? We can digest starch. We can't digest cellulose, right? We don't have the enzyme necessary to digest cellulose. And we can obviously digest glycogen, right? So glycogen is made up of 2 types of bonds. First is the most abundant type of bond. It is the alpha 1,4 glycosidic bond. Someone wiped this down with like a really sticky material. It's very strange. It's like the whiteboard is like sticking to my fingers. The alpha 1,4 glycosidic bond, which is the straight chain bond. And then we have the alpha 1,6 glycosidic bond, which is the branching bond. So this is the straight bond, and this is the branching bond. Now, what do we mean by straight and branching? Well, if we draw 2 glucose monomers next to one another, and this is where the person who commented on my video is going to start tweaking because I'm drawing these so quickly. So if you draw these next to one another, you can see that this is the 4 carbon, and this is the 1 carbon, and when these 2 bond with an O in the middle, that's a 1,4 bond. And when the 1 carbon over here binds to what? A 6 carbon, right? So when this, I guess we should go down, binds to a 6 carbon, like that, that's a branch. So you can see if we keep going like this, it's a straight chain, then this is a branch, and then you can continue the straight chain here and here, and it's a branch, continue the straight chain here and here. And what does that do? It allows for multiple positions where glucose can be broken off the glycogen polymer. And that allows for rapid mobilization of energy. And when does that happen? So the rapid mobilization of energy, that's a very key phrase, the rapid mobilization of energy, happens in fight or flight. Because epinephrine upregulates glycogen breakdown. And that's another high yield fact. Another key fact, high yield fact about glycogen breakdown is that epinephrine upregulates glycogen breakdown, which makes sense because you're mobilizing energy stores that you have inside your body, right? Technically, you may not need to know this, but I'll throw it out there. There is a little bit of glycogen inside your muscles, right? And there's a specific enzyme called myophosphorylase or muscle glycogen phosphorylase that handles the glycogen inside your muscles. And that is very responsive to epinephrine and very responsive to calcium as well, which makes sense because calcium release inside of the muscle cells means they're active, right? There's also a bunch of diseases that's associated with glycogen inside the muscles, and we're not going to get into that. And you'll get there. And then you'll cry about it like I did. So this is the basics of glycogen, right? And we have 2 things that we can do with glycogen. We can break it and we can build it. We can break it and we can build it. We can break it and we can build it. We can't really do anything else. You can't take it and move it somewhere else. You can't just like stare at it. Well, you could. But you break it and you build it, and you break it and you build it, you break it and you build it, right? So let's talk about glycogen breakdown, which is regulated by what hormone? I just said epinephrine, right? And? Norepinephrine, yes. What else? Glycogen breakdown means we're what? Low sugars or high sugars in the body? Low. And low sugars in the body means you're going to release glucagon. Here's an upper level question. Fructose 2,6-bisphosphatase, on or off when glycogen is being broken down? Think about it. Think through it. Take a second, on or off. Well, I have to think about it as well for 1 second. Hold on. Should be on, right? That's the conclusion I came to. And how did I come to that conclusion? Well, let's talk about what this does. Well, fructose 2,6-bisphosphatase takes fructose 2,6-bisphosphate, which is an activator of glycolysis, and deactivates it. Right? So in effect, it deactivates glycolysis, right? And when sugars are low, we want to deactivate glycolysis, so we want more of this, so this should be on. And that makes sense because glucagon actually turns it on. What's the other enzyme linked to this? Does anyone remember the name? Well, if it's not the guy that dephosphorylates that, it's the guy that phosphorylates that. So it's PFK2, not 1, 2. PFK2. Very good. Yo, my man. What's going on? You see how empty it is in here? It's so sad. I swear, there's this one time we had 20 people in the lecture, and I was like, damn, people really want to learn about the MCAT. And that was one time, and then the next week we had 3 people, and I was like, oh, never mind. But we got a strong 3.5 thousand people watching online. Shout out to you guys. So we're talking about glycogen breakdown, right? So when glycogen begins breaking down, we send out glycogen through the liver, right? Mainly. Most of the glycogen is stored inside the liver, blah, blah, blah. Everyone who fasted for Ramadan should know this, right? You get the little pain inside your stomach, right? So what are the enzymes that are important in glycogen breakdown? Well, first you have glycogen what? Glycogen phosphorylase. Yes, the rate-limiting enzyme of glycogenolysis. Glycogen phosphorylase, which I'll never forget. Because one time I was doing a UWorld question, and it asked me about the rate-limiting enzyme of glycogenolysis, and I said it was glycogen kinase because I'm stupid and forgot the name of the enzyme, and then I actually cried about getting that question wrong. It was really bad. It was a dark time. So I never forgot glycogen phosphorylase, which is going to cleave what? What types of bonds is this going to cleave? You have to specifically know this. Alpha 1,4, the most abundant bonds inside of glycogen, alpha 1,4 bonds. It's going to break that down. It's literally just going to Pac-Man the chain, right? But you're going to get to a point where the chain kind of looks like this. Here's a glucose and a glucose and a glucose, and they're on a branch. And here's another glucose and the glucose and the glucose behind it, right? This right here, this thing, where there's just a couple of glucoses left on the branch, this is called a limit. You don't need to know this. This is called a limit dextrin. This is called a limit dextrin. And the way I kind of think about this, when you get to medical school, they become really important because you look for these inside of people's blood when they have specific enzyme deficiencies, right? These are called limit dextrins. And the way you remember that is because this is the limit. This is how much glycogen phosphorylase can do without help, right? Because after this, it needs the help of what enzyme? Debranching enzyme. So debranching enzyme is going to come and cut that right there. Cut what bond? The alpha 1,6 bond. To liberate last glucose molecules. So it cuts the alpha 1,6 bond to liberate last glucose molecule. I know, I just did not write English over there. So that is debranching enzyme. Right? And then you have the UDP transferases and blah, 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 which you don't really know about. But there's one last enzyme that you need to know about. What is it? Glucose 6 phosphatase. Glucose 6 phosphatase. We're just going to take glucose 6 phosphate and turn it into glucose. And that is only found in the liver and the kidney, a little bit. Glucose 6-phosphate is also a really important enzyme in what pathway? Glycogenolysis and? No. We say these two things together. Glycogenolysis and? Gluconeogenesis. Glycogenolysis and gluconeogenesis. Why do we say them together? Because when you're breaking down glycogen, you want to release sugars. Gluconeogenesis happens at the same time. Happens at the exact same time as glycogen breakdown. We talk about them together. Also upregulated by glucagon, also upregulated by epinephrine, also upregulated by norepinephrine. Gluconeogenesis. Someone explain to me why, and this is a good little tie-in here, someone explain to me why gluconeogenesis is increased in times of stress. Yes, but dig a little deeper. So when you have fight or flight, right, your muscles are what? They're really active, correct? So they're breaking down like stuff into what? Sometimes you go into anaerobic respiration. You produce lactate, and lactate can be circled back into the liver to produce pyruvate, and you can make glucose from pyruvate using gluconeogenesis, right? So it makes sense that when we have fight or flight activated and we're working out a lot and there's a lot of anaerobic respiration going on, the lactate levels go through the roof, that the gluconeogenic pathway is turned on to accommodate the influx of lactate. What is that cycle called? The Cori cycle. Very good. That is called the Cori cycle. So just as a side note, gluconeogenesis, right? Which, remember, what are the important enzymes? So we have pyruvate carboxylase, PEP carboxykinase. Also, what does PEP stand for? Phosphoenolpyruvate. So this is phosphoenolpyruvate carboxykinase, PEP-CK, right? Pyruvate carboxylase, PEP carboxykinase, fructose 1,6-bisphosphatase, which is the rate-limiting enzyme, correct? And glucose 6-phosphatase. These overcome one irreversible step. These overcome the next one, and these overcome the next one. That overcomes the next one, right? Okay, that's gluconeogenesis. And gluconeogenesis can use pyruvate and turn it into glucose. But what can turn into pyruvate? Amino acids can turn into pyruvate. Lactate can turn back into pyruvate. Right? And right here, when it's lactate, this is called the Cori cycle. And when it's amino acids, most often they'll ask you about the alanine cycle. Most often.
Did you explain the alanine cycle?
No.
Oh.
I will not explain the alanine cycle. I'll not be doing that. That has been thrown out of my mind. You also don't need to know it that well. Okay. I'd say the Cori cycle is much more important. So exciting. That's glycogen breakdown. Glycogen synthesis is stupid easy. Once you know glycogen breakdown, glycogen synthesis is really easy. All right. Glycogen synthesis. Okay, so number one, it's increased by insulin, right? Number two, what are your enzymes? You have glycogen, synthase, and branching enzyme. This does the alpha-1,4s, this does the alpha-1,6s, just the opposite of what we said before. Right, so this is the antagonist of glycogen phosphorylase. This is the antagonist of debranching enzyme. Makes sense. Branching enzyme is the antagonist of debranching enzyme, right? And you have to know that it starts from a little granule core. There's a little core, glycogenin. That's a little protein in the middle of the core, and everything grows off of that core. Kind of it. You don't really need to know much else. How does glucose get into the liver? Insulin dependent, insulin independent. Huh? GLUT2, is that dependent or independent? Independent. All right, so let's talk about insulin dependence, right? And the biggest ones you need to know are GLUT2 and GLUT4. And you only really need to know one example. GLUT2 is the liver, and GLUT4 is the muscles. Think about it this way. If sugars couldn't get into the liver without insulin, the insulin wouldn't know how much to react to the sugars in the body. Why? Because you have glucokinase here. You have fructose 2,6-bisphosphate being made here. That's going to regulate the amount of glycolysis that goes on. You have the glycogen here. You have gluconeogenesis here. So the liver needs to be able to sense, hey, how much glucose am I actually taking in at any given time, right? The GLUT4 is less sensitive, right? It's like, yo, I'm not going to let you in until I know I'm supposed to, right? So this is responsive to insulin. Insulin opens the GLUT4 transporter to allow the glucose into the muscle cells. Whereas this is insulin independent. However much glucose comes in, you're going to allow it in. Whore, conservative. Right? She's a 2. She can't be really picky. No, she's a 4, you know, like, And neither of them are good, but, you know, she can be a little more picky than she can. He can be a little more picky than he can. Don't fucking cancel me, all right? Yeah, and then we have GLUT1 as well. I believe that's in the nervous system, the brain, the eye, things like that. GLUT1 is also insulin independent, right? Because if it were insulin dependent, you'd fucking die. You'd go into a coma, your brain wouldn't work. And then also, one more very high yield place where glucose can get in without insulin is what? Red blood cells, right? The red blood cells. The red blood cells need to be able to use glucose. But what can they not do with the glucose? They can't do what? Oxidative phosphorylation. They can only do, and this leads me into my next, you see the segues here? Oh, you see the segues here? Why do we make a differentiation between oxidative phosphorylation and whatever else the next thing is? What is the next thing? Substrate level phosphorylation. Wow, look at these segues, man. It's just, I'm ascending right now. I'm peaking. All right, let's talk about oxphos and substrate level phosphorylation. Oxphos, right? Like this is the way I want you to think about it. Oxidative phosphorylation is just what happens in the ETC. That's it. The ETC is oxidative phosphorylation. You are using oxygen, and the energy released from the reduction of oxygen by its electrons powers the motion of ATP synthase, which allows for the inorganic phosphate to be added to it in the phosphoride anhydride bond, and you get the production of ATP. That is oxidative phosphorylation, because the oxygen oxidizes whatever's in the complex, right? It gets reduced. It's oxidative phosphorylation. Substrate level phosphorylation. When we say substrate, you say the word enzyme substrate, right? Shut up. Enzyme substrate. The substrate is definitely a sub, though. It's always just getting slapped around. So enzyme substrate, enzyme substrate, enzyme substrate. So when you use an enzyme like hexokinase, glucokinase, fructokinase, what else? Glycogen phosphorylase, right? Phosphofructokinase 1, phosphofructokinase 2. All of these are substrate level phosphorylation, right? Because you have a substrate and you phosphorylate it using an enzyme, a kinase or a phosphorylase. Don't be me, and 3 weeks after your exam, confuse a phosphorylase and a phosphatase. Don't do that. They're complete opposites. right? So that's substrate level phosphorylation. Let's talk about cholesterol for a second. Nah. I was gonna make a joke about me being brown, like having high cholesterol run my family, but you know, everyone's heard that one before. All right. Man, this sucks.
Yeah.
This is the worst.
What happened?
Oh, you didn't come in. Tuesday's my last lecture because I'm on call on Thursday.
Thursday?
So I'm not going to be here. I'm on call from 5 to 10 p.m. on Thursday. I'm actually working the psychiatric emergency room, so I will not be here on Thursday. Can someone let him in? I think he went to the wrong door.
Other door.
Yeah, this sucks. I've been here since August. How many months is that? We took off December, and I took off for Ramadan, so that's 2 months that I took off. How you doing, man? What's going on? So we did. Well, technically I did September, October, November. I took off December, then January, February, March. March I took off. And we did like half. We're doing like half of April. It's like 5.5 months. Oof, 5.5 months, and you guys aren't tired of me yet?
And no jelly ranchers.
Oh, no Jolly Ranchers now. I mean, that was in the beginning when I had to get you guys on my good side and everything.
He was.
And now I got to bring him back. Seriously. But this sucks. No, it really does. Because it's just like, I don't have anything else to teach you. I feel like this is what teachers feel like at the end of the school year. It's just like, I've got to tell you guys everything. Like, I'm telling you everything I know. Except lab techniques, which we'll be doing on Tuesday. We'll be doing separation and gel electrophoresis after I rewatch my Khan Academy videos and learn how to teach it to you guys again. Khan Academy, the GOAT. He's probably a lot better at teaching than I am, so if you want to go check him out, check him out. Let's talk about cholesterol. So we can ingest cholesterol by itself, of course, right? They tell you egg yolks are high in cholesterol and red meat is high in cholesterol. And we can also do something known as a de novo synthesis of cholesterol. And we build up cholesterol 2 carbons at a time. From what? What do we use in our body? Well, yes, terpenes, right? We'll get to that. What's the other thing? Terpenes and what else? Isoprenes, yeah, isoprene units. But we build it up 2 carbons at a time. What in our body do we use 2 carbons all the time? We use it all the time. Acetyl-CoA, right? So we turn this guy into that guy. But also they want you to know about this guy. This guy. Is it like this or is it like this? Is it just this? Is there a double bond?
Yeah.
There is, right? All right. And I always got confused by this. I never really understood because we said we make it from 2 carbons, and then you got 1, 2, 3, 4. What the fuck? Why is there 5 carbons here? So that doesn't really make sense to me. But they want you to know that somewhere in the cholesterol synthesis pathway there is a isoprene unit. And that cholesterol can be built up of back-to-back-to-back isoprene units. And then when you build up isoprene units together, they can make these things called terpenes. Now, I'm a huge nerd, right? Massive nerd. So I used to do a lot of independent research on drugs and how they affect the human body and how they affect the human mind. Terpenes are actually very, very high in what kind of drug? THC. Yeah, so marijuana. Marijuana is very high in terpenes, and terpenes can also cause the sort of euphoric, sedative, and feel-good effects of marijuana, right? I think, is it terpenes or is it, there's that other thing in wine. I think it starts with a T as well. That thing that makes wine, tannins, yeah. So wine has tannins, and then marijuana has terpenes, right? So I don't drink. That's not why I know that. I'm just very, very, very interested by alcohol culture. I think it's super interesting how we built up an entire culture around subtly poisoning ourselves. So that's very interesting to me. If you want my advice, don't drink. But I know you won't take my advice unless it's something that's on the board. So. Don't drink, smoke, fuck, drive under the influence, kill me, cancel me, or die. Those are all things you can't do. You can't drink, you can't smoke, you can't fuck, you can't drive under the influence, you can't kill me, you can't cancel me, and you can't die. If one of you dies watching this video, I swear to God.
Oh my god.
Well, they were distracted about driving.
Do you guys ever just like, are you guys ever just like driving? So whenever I drive back to Long Island, this happens. I'm like driving, I drive past my old like elementary school, and it's like I take a right onto the road and I go down towards my house. And like the last 0.5 miles of driving, I was like, that just didn't happen. Like I don't remember driving the last like half mile to my house. I was like, I could have like ran over 7 children and not known it, like, because it's just so muscle memory and so zoned out. And like, I know where the red light is, I know how long it is, I know where the stop sign is, I know there's a hill. After the hill, you gotta slow down because there's a 50 mile, and then you gotta take a right, and then you gotta pull into the driveway. And I was just like, oh my fucking God, I could have killed 9 people on the way here.
You're stuck in the
It's so bad. It's so bad. Yeah, but no one knows where I live. No one knows my hometown.
But once we figure it out,
I actually don't put my hometown anywhere. I don't put, not anymore at least. It might be in like one place, which I'm probably gonna wipe it from now that I put all this on video. But no one knows my hometown. Good luck searching every town on Long Island to find an elementary school with a hill and a right turn next to it. Good luck. That's why like people are like, oh, don't tell people where you live on the internet. And people are like, oh, where are you from? I was like, New York City. Good luck finding the brown guy in New York City. Like, have fun. So cholesterol is obviously the backbone of our steroid hormones, right? Estrogen, androgens, right? Progesterone, cortisol, things like that. And then also, cholesterol is packaged into what? Huh? Lipid rafts, right? This guy's been doing his fucking Anki, bro. This guy's been going hard on the Anki. And then you got like the LDLs, the VLDLs, which you don't need to know too much about. But the one thing I do want to comment on is what LDL is lethal, HDL is healthy, right? HDL is the scavenger lipoprotein. LDL is the lethal lipoprotein. It's going to deposit cholesterol all over your body, and that's how heart attacks happen, right? Heart attacks, peripheral vascular disease, carotid aneurysms, strokes also happen from there. You'll learn all about that when you're in medical school. That's good, everything like that. Also, big thing is that you don't really absorb cholesterol if you don't absorb fats properly. So there's a lot of diseases where you don't absorb fats properly, right? Like Crohn's disease, right? It can cause fat malabsorption, right? And diarrhea can cause fat malabsorption. Things are just moving through your digestive tracts too quickly because there's actually a very small window within which you can absorb fats inside your digestive tract. So when you don't absorb fats, you get a relative deficiency in your cholesterol, which is fine because you normally have a buildup of it anyways. But what vitamins do you end up getting deficient in? ADEK, right? Because they're the fat-soluble vitamins. So A, D, E, K, right? I want you to think of all these things together. So the ADEK, and then the cholesterol, and fats. I want you to think of all of these 3 things as coming in sort of panel, and all being shunted into your body in the same time, at the same time, in the same way. Inside chylomicrons, inside lipid rafts, inside of like lipoproteins, blah, blah, whatever it is, right? You should think about it like that. Okay? Yeah. Here's a DNA double helix. Horrible segue. And eventually, it does this. What is this? Histones. Yeah, these are histones. But specifically this thing, the histones plus the DNA wrapped around it, what's it called?
optimer. Histone optimer.
No. Heterochromatin is when just the DNA that's compressed like that. Yeah. This is called a nucleosome. It's called a nucleosome. I remember there's 8 of these guys, and then an H1 rod that runs up and down next to it, right, that the histones kind of anchor to, right? You don't need to specifically know that. But these like H1, H2, H3, H4, stuff like that. And then the DNA is wrapped around this is what, euchromatin or heterochromatin? Heterochromatin, and then it's euchromatin when it's free. So euchromatin is free and euphoric, right? It's very happy. Heterochromatin is you know how straight guys are like really, really like averse and don't want to talk about being gay? The straight guy's like super closed off and like, oh my God. Ugh. So that's heterochromatin.
Oh my god, the most gay way to
Oh my God. Ugh. I'm straight, by the way.
Yeah, okay.
That was the punchline right there. I think I dinged that one. I've heard that so many times in high school because I grew up on Long Island, right? So I grew up with all that, like the jock football players, like, ugh, like they're just like, bro, I'm so straight, like I get so many ladies. I'm the football captain, blah blah blah. Meanwhile, I was on the cross country team where like we wore shorts that were like 1 inch off from our fucking thighs and ran around and commented on how nice everyone's legs looked while they ran. Gay! Anyways, histones, right? So we need a way, because our DNA normally exists like this in the chromosomes. When you see DNA in chromosomes, it's nucleosome, 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 just heterochromatin, 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 just super packed in on itself. But we need a way for genes to be able to be transcribed when we need them to be. So we need a way to be able to release the DNA from the histone. Well, what's associating the DNA to the histone? There's an interaction that's associating the DNA to the histone. What is it?
Lysine.
Huh?
Lysine.
There's lysine in the histones, right? There's lysine, and there's lysine, and there's arginine, right? And what is that binding to in the DNA? The backbone. What in the backbone? Phosphates. So the negatively charged phosphates are attracting the positively charged lysine and arginine. And we have to somehow interrupt that interaction. So what do? We acetylate the histone, right? So if we were to look at lysine, don't quote me on the number of carbons. I always fuck it up. You guys have to know it. I don't. Do as I say, not as I do, right? When you look at lysine, this guy can actually attract this guy. It's going to track like that. But what if we did this? Is there any interaction anymore? If anything, there's very little interaction. Why? Because you see how we acetylated that, and now there's a nitrogen next to an sp2 position. So it actually kind of looks more like this. And now that negative terminal is going to push that phosphate away. So when you do that all down the histone, it's going to push the DNA away from itself. The DNA is going to get exposed, and the transcription factor is going to be like, that's our fucking guy. There he is. Right? And then you're going to start transcribing. Transcribing. What set of hormones act specifically as transcription factors? Steroid hormones. Why? Because they can enter the cell, they can bind to intracellular receptors, and then localize to the nucleus and affect transcription. Right? Transcription factors. I had a great time. I'm just waiting for you guys to finish writing. Okay. There's one more thing. So we talked about histone acetylation. And there's one more thing we have to talk about. There's DNA methylation. Does it do the same thing or the opposite thing to histone acetylation? Opposite, right? Because let's imagine you were a hiker. You're very happy. You got your little hat on, right? You got your backpack. I don't know why you're naked, but you're hiking, right? You got a little backpack. You got a little bottle of water. Maybe you have a cat with you. Fuck ass looking cat. He's also really happy. He's just happy to be here, right? You're walking. And you're walking on this trail. You guys kind of see where this is going? You're walking on the trail, and you're trying to get all the way over here, right? This is it. You're like 0.4 feet. You're like 0.4 times 10 to the negative 16th meters from your destination, because you are microscopic. And you're walking along this path, and all of a sudden, boom, there's this giant log sitting in your way. Are you going to be able to walk through that? No. Your cat's going to turn his ass around. He doesn't want to climb over that, right? You have to do a lot of work to get over that. This is the methyl group on the DNA, right? It interrupts the ability for transcription factors to walk across the DNA. So when you have a DNA strand like this and you just methylate it, you can't read it anymore. It's like a different language. So if I went into a textbook and I replaced every single other letter with like every single other letter, like you wrote out a whole thing, every other letter I put in the letter A, would you be able to read it? And no, it's gonna be hard as hell. It's gonna be really difficult to read that. Look, it says Brooklyn College right there, right? BARAOAOAKALAYANA. Like, who the hell is gonna read that? So that's what the methyl group does. A. Like, well, not actually A, because that means something in biochemistry, but you know what I mean. So you can't read it anymore. You can't transcribe it. You can't make the gene happen. You can't produce the protein. You can't produce the mRNA. So DNA methylation is a way of decreasing transcription, and histone acetylation is a way of increasing transcription, right? Here's a very sort of high-yield fact. I'm not going to talk about COVID because, like, I should, and you guys all know where that conversation is going, but I don't want to get, unfortunately, political on my YouTube channel because who knows, maybe some people watching have political ideas about the vaccine and things like that. But for 1 second, let's talk about mRNA. Why is mRNA safe to inject into the body? Because it doesn't what? It doesn't last. It's going to destroy itself. Why? Why is DNA relatively stable? The second carbon. What about the second carbon? Right. So if we look at the backbone of a ribose molecule, because RNA is ribonucleic acid, and then DNA is deoxyribonucleic acid, and specifically it's 2' deoxyribonucleic acid, right? So if we look at this, I'm mad close to this board right now. It's like making me uncomfortable. It starts whispering to me sometimes, telling me I'm stupid.
You what section?
No, not like that. Wait, let's see if I remember this. All right, someone who knows these better than me, quote me on this. I know there's something over here. Right? Is that correct?
It looks right.
If it's wrong, sue me. Send me a Venmo request for 1 cent if I'm wrong. I'll actually fulfill it. Imagine like 30,000 people send me a Venmo. I'd pay $300 for writing adenine wrong on the board. All right. So this is adenosine, but the RNA version, right? So you see right here, this oxygen can become 1, 2, 3, 4, 5, 6. And it can attack right here on that 5 position, right? It can loop in on itself, attack the 5 position, and kick off this super stable triphosphate. Because when you kick it off, that oxygen is now allowed to resonate with the rest of the phosphates over here. You can kick that off, and then what's that going to do? It's just going to close that ring. It's just going to completely close that ring. So we can kind of see what it looks like, right? So we're going to have oxygen. Let's see if my drawing skills are still good. Right, so we have oxygen, and we have OH right here, and then we're going to have a ring, and the ring is going to be 1, 2, 3, 4, and then 5. And it's kind of going to be like a bicyclo compound. So I think this guy's going to come this way, and then this can go like that. So you can see how it's sort of like a bicyclo compound where you have this 5-member ring over here, you have the triangle in between them, and you have the other side of the bicyclo like this. So this, it doesn't look fun, but it's stable because it's 2 5-membered rings, right? And they're merged by this bridge right here, right? So that can happen. And what that does is it kicks off this very stable triphosphate, which is just going to get attacked by other things and get broken down, right? So the RNA inside of your body, it can loop in on itself, break itself, and destroy itself. So it's metastable. And at some point in the lifespan of the RNA, that's going to happen. It's going to happen. It's just going to destroy itself. And then that's going to get broken down, and this is going to get broken down, and it's gone. That also helps us to regulate gene expression. Because when the RNA gets broken down, you're not going to continuously produce protein from it, right? However, now this is where we separate the 520s from the other people. This isn't really a 520 concept. This is more of like a 515 concept. What are the 2 modifications we make to RNA in order for that not to happen, and where do they happen?
Isn't it the 5' cap?
The 5' cap, right? And the poly A tail. All right, you got most of it. 5' cap, which means this is what? Poly A tail is on what end? The 3'. The 3' poly A tail. What is, there's one more fact you're forgetting. What is the cap? Mm-hmm. Is it? Yeah, yeah. No, you're right. So the way I learned it, like back when, I think you're correct, it's 7-methylguanosine. It's basically GTP. It's a modified GTP molecule, right? So 5' guanosine cap is a 3' poly A tail. Very good. So you guys know this very specific niche piece of information. However, how are they going to ask you about this, right? They're going to ask you, a protein has 33 amino acids, right? They want you to estimate, right? You guys have probably seen a question like this before. You know exactly what's coming. You know, because they ask this all the time. A protein has 33 amino acids, right? And they want you to say, which of the following best describes how many nucleotides were inside of the original mRNA sequence? 99, 102, 99, 102, less than 99, or greater than 102? It's clearly not less than 99. Why?
Because you have a lot of A's.
But why specifically 102?
Introns.
What? The stop codon. So it's in 33, makes 99 coding amino acids, including the start codon, which is usually methionine, plus the stop codon, which is 3 more, plus the poly A tail. So it's far more than 102. But how do they derive the number 102? I just told you. But some people, they fling, they're just like, oh, 99, stop codon, 102. We're talking about mature mRNA. Yeah, we're talking about mature mRNA. But that's a good thing to talk about, right? So introns and exons. Exons are expressons, right? So exons are expressed. And that's just alternative splicing. So what can happen is you can have a gene that looks like this. E1, E1, I1, E2, I2, E3, I3. Sometimes I like to fuck with my students, and I'm just like, yeah, bro, like an E3 reaction. And they're just like, wait, what? There's more. So I can take out everything here, and I'll just have E1 and E4. Or I could take out like something in the middle of E1, E2, E3, E4. Or I can have E2, E3, E4. Or I can have E2, E3. Or I can have E4, E2. Or I can have E3, E1. Like I can have a multitude of things going on depending on where I splice, right? What are the things called, the enzymes called that splice RNA? Spliceosomes. Oh my God, so crazy, right? And now just some random facts that come to mind when I think about biochem. What does topoisomerase do?
That's the one that prevents it makes it straight with the DNA.
Yeah, so it sort of holds down the part that's being elongated, that's straightening out. What specifically what it does, it prevents torque on the replication fork. So what's happening is that as the replication fork is turning, as you're straightening out the DNA, you create a lot of force against the replication fork that may cause the DNA to break. It may shatter, right? So topoisomerase kind of holds it down and allows it to unfurl properly, right? Okay. DNA ligase. Everyone knows what DNA ligase does, right? It just comes in, like spits down a little nucleotide right there and spits down another nucleotide. It just fills in the gaps, right? It's like glue, right? What are the things that it's filling in? What are they called?
DNA bricks?
No. They're called Okazaki fragments.
Oh shit, yeah.
They're called Okazaki fragments. What are Okazaki fragments? So basically, you have a leading strand, you have a lagging strand. DNA synthesized in what direction? 5 to 3, right? So the replication fork going that way, and this way, here's topoisomerase holding it down, right? 5 prime to 3 prime, and then this is anti-parallel, so this is 5 prime to 3 prime, right? So we can build this way in a continuous fashion, which is called the leading strand. And then this has to be built like boom, 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 boom. And then ligase has to spit a nucleotide into those slots right there. And those little fragments are called Okazaki fragments, after the person who discovered them. Probably. I don't fucking know. I didn't discover anything. Okay. Thinking. Because I've taught you everything. Because I've done it all. Throw something out there. Biochem related. See? You can't even think of anything because I've said it all, baby. Biochem is my strong suit, so.
You could draw another cat.
I draw another cat. Nah. I don't want to torture you guys. People back home? I think the rest of it is honestly just like lab techniques and things like that.
Did you go over like homeostasis, like you have chapter 12 or?
Yeah, I mean,
The video's good though.
there's not much really to say there. You know what I mean? Like it's just thermodynamics, things like that. I talked about those. What's it called? Equations so many times. I wanted to do amino acid titrations. I just don't think it's high yield enough for me to cover, and I'm also not that great at them. Hemoglobin? No, because I have so many videos that go over hemoglobin. I have way too many, way too many. But I think the biggest thing was what we recorded last time in the I think it was Bio Rapid Review 2. I went over hemoglobin. So I'm also just shot. I can't do any more hemoglobin. Like, hemoglobin is just the bane of my existence now.
Do we have some of the different like DNAs, like DNA A, B,
Nah.
H?
I don't think so. The question was, do you need to? This is something people commented. They were just like, you never repeat the question when people ask them. So the question was, do we need to know the different DNAs? And the answer is no. I didn't know them. That doesn't mean you shouldn't know them. If you can know them, sure. If you don't want to know them and you think, can I still get a good score? I'm the living proof. Yeah, guys, I think that's it. And next time micelles? Okay, let's go over micelles. So micelles are basically when your body takes triglycerides in, right? Your body is going to take these triglycerides and cleave off these portions right here and make fatty acids, right? So, did I do this wrong? No? I think, yeah. I'm tripping. All right, so it's going to chop those off, and it's going to make fatty acids. Now, these fatty acids are pretty insoluble inside of the bloodstream, right? So what's going to happen is they're going to line up almost like phospholipid bilayers do, right? So you're going to make a sort of bilayer where all the polar ends are going to line up on the outside, and all the non-polar ends are going to go inside, right? And what this does is it allows for a non-polar environment to be made inside of the micelle. And what does that allow for? It allows for the absorption of things like, what did I say before? ADEC, cholesterol, and fats. So micelles move around like this, and they can selectively drop off things after being, assorted with lipoproteins, things like that. And you'll learn all of that when you do biochemistry in medical school. It's literally the worst thing in the world. I absolutely hate this stuff in medical school. And I hate it now too. But that's a micelle. Just for the hell of it, let's do one last thing before we call it a day. Let's go over the names of the vitamins. Who remembers the names of the vitamins? What's vitamin B1? Thiamine, right? Vitamin B2? Riboflavin. Vitamin B3? Niacin. Vitamin B4? There is none. B5? Pantothenic acid. B6? Yeah, pyridoxine or pyridoxine phosphate, right? B7, biotin. B8, there is none. B9, folic acid. There's no B10, there's no B11, and B12 is cyanocobalamin, right? What are they all important for? Vitamin B1 is actually a cofactor in a lot of reactions called TPP. Specifically, it's used for alpha decarboxylations, right? Do you guys know pyruvate kinase and alpha-ketoglutarate dehydrogenase? Both of those require alpha decarboxylations of a specific substrate. And whenever that happens, you need TPP. Technically, you need a lot of different cofactors, and there's a set of enzymes that use these cofactors. It's called tender love and care for Nancy. So it's TPP, lipoic acid, niacin, so B3, and care, CoA, and then for Nancy is FAD and NAD. Yeah, so it's like that. So it's like tender love and care for Nancy. So TPP, lipoic acid, CoA, FAD, NAD. And the enzymes that use these are, am I okay? Okay. The enzymes that use these, I'm telling you, they did something to the board, dude. It's not the same. So the enzymes that use these are pyruvate dehydrogenase, right? Alpha-ketoglutarate dehydrogenase, alpha-keto acid dehydrogenase, which you guys don't need to know about. It's important to a disease called maple syrup urine disease. It's the real name of the disease. because people's urine when they have the disease smells like maple syrup. And there's one more. Enzyme. I forgot what it is. But you'll realize that in all of those things, right? So if you have pyruvate, and then if you have alpha ketoglutarate, oh my God, wait. Wait, no, I have to go from citrate. Sorry. I can't just do it off the top of my head. I'm not smart enough. Okay. Citrate, isocitrate, alpha ketoglutarate. There we go. Alpha ketoglutarate, right? You'll see carboxylate alpha to a ketone, carboxylate alpha to a ketone. So when that carboxylate has to fuck off and go away, we use TPP, which is thiamine pyrophosphate. Riboflavin, flavin adenine dinucleotide, FAD. Niacin, NAD, nicotinamide, right, niacin. And then B5, pantothenic acid. What does it do? Nothing you really need to worry about. B6, neurotransmitters most of the time, right? And a little bit of heme synthesis as well. B6 is really important to heme synthesis. B7, hair, biotin, right? B8. Also, B7 is important for decarboxylation reactions that aren't alpha decarboxylations, regular decarboxylations, right? B8, there's none. B9, pretty much fucking everything, but really specifically, neurodevelopment and neurologic maintenance, right? And babies love to eat up mom's folic acid, so you have to take folic acid supplements. And then B12, also neurologic stuff and blood stuff and amino acid stuff and nucleotide stuff and homocysteine and methionine and SAM, and it does a lot of stuff, and you really don't want to be deficient in B12. I will see you guys one last time on Tuesday.